Hello, everyone. Welcome back to my Science and Religion series. And so, as we continue and looking at different topics, both in the scientific community and in the Bible, we are currently going to be looking at chimps and humans. And do they have a common ancestor? So, if you don't know, my name is Dr. Christina Sweeney Hillesheim. And so, now let's just dive a little bit deeper into kind of macroevolution. So, if we want to start talking about macroevolution and do chimpanzees or apes and hominids, meaning us humans, Homo sapiens, if we have a common ancestor, it would be kind of a good place to start with what is our kind of closest. So maybe not going all the way down kind of the tree if we're thinking about it from a Darwinian perspective, but thinking what is our current most similar or most like skeletal structure. So the fossil that most resembles a human skeleton is Cro-Magnon, and it was first discovered in a cave in Bordeaux, France. And there has actually been, even though that was the first place that it was discovered, there's actually been species found across Europe, Asia, and the Americas. So it's not isolated just to one geographical location. And there's several similarities between the Cro-Magnon fossils and the human skeleton, Homo sapien. And so the similarities are the hip bones. They're shaped in a way and for standing erect. The facial bones are very similar, and the shape and the size of the brain cavity are very similar, which is one of the things that's very different compared to kind of other similar human-like skeletons. So this skeleton first appeared in the fossil record between 50 and 100,000 years ago. And so that's when it first appeared. And just to give you an idea, about 10,000 years ago, there were so many of these fossils that that were found at that point that we would be able to fill a museum. So it wasn't just like, oh, we had a few of these and there's not that very many. We actually have a lot of these fossils that would be able to fill up a museum of just these fossils. Just to give you an idea of the magnitude of the fossil record for Cro-Magnon. So you might say, okay, so there's Cro-Magnon and the modern man. So what is a biblical take on this? How does the Bible perceive this issue? So there's actually no biblical problem with human-looking creatures predating Adam. And so the Bible actually differentiates between what would be human and what would be animal-like creatures. So the Bible defines a human as an animal with a neshama. And so, you have all the different creations. So fish, you have insects, reptiles, and then you have chimpanzees and apes, and even Cro-Magnon. And the way that the Bible differentiates all of these from mankind, from Adam, human, modern man, would be this neshama. And so this neshama is that spiritual soul. And this is seen in Genesis 2:7. So the idea is that having a soul is what differentiates you from the other group. And so there were animals with human shapes, but lacking the neshama. And so this is how, from a religious perspective, it would be kind of okay and understood. Is that kind of the dividing point would be if it has a soul or not is on one side. If it doesn't have a soul, it's on the other side. Is how to think about it. So you might be also asking yourself, okay, well, maybe you haven't heard of Cro-Magnon, but you've heard of Neanderthals. So what is the difference? So it's interesting though that Cro-Magnon fossils overlap in age with the more primitive Neanderthals. So you could kind of think about it as like Neanderthals, Cro-Magnon, and then humans is how it's kind of our modern man is how it's kind of illustrated in the Darwinian explanation. But interesting enough, the fossils, so if it were Neanderthals and Cro-Magnon, then there shouldn't be an overlap. One should go into the other, but there's actually an overlap in these two fossil groups. Now, to give you an idea about Neanderthals, here's a picture. They kind of resemble the usual pictures of what we think of cavemen. So, cavemen are what we think of with Neanderthals. They have a protruding brow and kind of a sloping forehead. But this discovery, this overlap, actually countered the theory that Neanderthals developed or evolved into Cro-Magnon. Because if one evolved into the other, then you should see either one going away, the other one staying, and some kind of intermediate. But since they both existed at the same time and there's overlap in this in the fossil record, this kind of provides no evidence that Neanderthals were a precursor for Cro-Magnon. And there's no evidence of an immediate ancestor to Cro-Magnon. So we're not really sure how they evolved or where they came from, if that were the process. So what we want to do is we want to actually try to think about, well, where did Cro-Magnon come from? So in the previous slide, I said that it was thought of that Neanderthals kind of evolved into Cro-Magnon. But because of that overlap, that the Neanderthal couldn't be the precursor or the previous ancestor to Cro-Magnon. So, and we don't really see any evidence for it. So what could be that precursor? And so that's what we want to kind of try to figure out. So we need to focus on the moment in which it's theorized that a common ancestor diverged into two populations. So to really figure this out, we kind of need to go back a little bit more. And so what we want to do is we want to go back into the line here where it's thought to have branched off and one became what we think of as modern day chimpanzees and the other as Homo sapiens. So we want to kind of go back where this common ancestor was and look at this separation, this breaking in the kind of tree here. And it is believed that this divergence occurred 7 million years ago in Africa. And I'll explain why. So, for about millions of years, the topography or the landscape of Africa really posed no barrier to free movement. So, everything that was living in Africa could intermix, intermingle, interbreed if possible. There was nothing really stopping all this life from just kind of coming together. There was no physical barrier. But all of this changed dramatically 8 million years ago. So 8 million years ago, there was a change in the topography in Africa that then allowed for there to be this barrier preventing this intermixing. So there are shifting tectonic plates that are produced along the Afro-Syrian Rift. So you can kind of see these plates in this picture here. And this is just a zoom out of the picture here of the African continent and the Middle Eastern area here. And so here, there's a ridge that's running south to north through Africa and continuing to Aqaba Ilit, Jericho, and then onto Syria. And so what happened is the African portion of the ridge kind of isolated the eastern third of the continent from the western two-thirds. So you've got one third here in the eastern part, like right here, from the two-thirds of the western part. And so what happens is this became a natural barrier. And so this natural barrier, what happened was now animals that were in the east could no longer really mix well with the west and vice versa. So now we don't have easy mixing and we don't have interbreeding. So now they're kind of isolated. And so this tectonic shift was able to establish what we call genetic isolation. And so genetic isolation is one of the traditional prerequisites for speciation. And so this is really important because if we want a species to change, they need to have some genetic isolation. So now we have some animals now on the left, on the western side, and some animals on the eastern side, and now they can't intermix. And so both of these two groups are going to be genetically isolated. Now, because this ridge from this tectonic shift was created, it was actually really steep. And so it actually changed the environment that was being experienced. It changed the flow of the air and the weather patterns across the continent. So the western region actually retained kind of the same environment. It retained those high levels of moisture that it had, arboreal vegetation, and so it didn't really have any new environmental challenges. And so this is thought that that common ancestor, they're the ones that kind of went and evolved into modern chimpanzees. So chimpanzees came from the western sides of evolutionary change. On the eastern region, we actually had a reverse pattern of the air circulation. Because of this ridge, the air pattern changed, and now this air was now stripped of any moisture entering the region. And so what happens is this landscape became less humid, less moist. The forest started to dwindle away, and instead of it being kind of more of this arboreal vegetation, the landscape is now going to change more to savanna grass with occasional trees, kind of seen in the picture in the background. So it's thought that this new environment actually favored creatures that walked upright versus creatures that would live more in the trees, like a chimpanzee. And so, because of this, those tree dwelling primates then would kind of evolve into upright primates, our first hominids, and then that's what eventually evolved into Cro-Magnon. And so this is where you get that separation of modern chimpanzees and then Cro-Magnon. Now, what do we need for this evolutionary change to occur? So we need a model for the genetic changes that might allow for a tree-dwelling ape to become an upright Cro-Magnon. Now, humans actually differ genetically from the chimpanzee by 1 to 5%. And so what we're going to do now is we're going to look at a kind of a mathematical analysis of what would be needed if we wanted to kind of go into this evolutionary process. So we want to see, do we have enough time and how long would we need for this evolution process to occur? So going from that common ancestor into, either a chimpanzee or Cro-Magnon. So our human cell contains approximately 3 billion nucleotide bases. And so remember these nucleotide base pairs are A, T, G, and C. Those are the kind of short notation for them. And they run along the backbone of DNA. And then they match up based on their pairing. Of these 3 billion nucleotide base pairs, only 3% are actually expressed in the coding of proteins. So the other 97% of the DNA seems to be inactive in coding for proteins, but that doesn't mean that they don't have any purpose. We've actually found this is a whole nother conversation regarding junk DNA and does that DNA actually have purpose? And it has been discovered that it may not be involved in coding for proteins, but there are other very important functions for the other 97% of the DNA. But we're just going to focus on that 3% because what we need for us to have evolution is we need a change in the genetic code related to proteins. So thus, to create a chimp or Cro-Magnon from that common ancestor. So remember, we're looking at here's the common ancestor, and then we have chimps, and then we have hominids. Specifically, we're going to look at Cro-Magnon. And so what we need is you only need to change the active part. So we only need to change that 3%. So remember I said there's 3 billion nucleotide base pairs. So this is the 3 billion right here. But we only need to change the 3% that is the active. And so when you do 3% of 3 billion, you get 90 million 90 million bases that we need to actually be able to change. Now, if we assume that humans differ from chimps by 1%, so remember I said it's 1 to 5%. And so one of the things that we're going to be doing throughout this model is we're actually, if we have like a range 1 to 5%, we're going to pick whatever number actually favors evolution. So we want to try to do the math where it is kind of the best possible solution for evolution to occur. So picking the best situation, best numbers, when we're doing our math. So instead of 1 to 5%, we're going to try to make it the closest and the least amount that we need to change. So we'll only need to change that 1%. So we only need to change 1% of that 90 million bases. And so 1% of 90 million is approximately 1 million. or 900,000. And so that's what we need to change is approximately 1 million differing bases. So is it possible to have 1 million point mutations in DNA during the 7 million years available? Because remember the reason that 7 million is because 8 million years ago, that's when we had the ridge that formed in Africa, and we saw the first fossils 7 million years ago from our Cro-Magnon. And so we need to be able to have that. That's when we had the genetic isolation. And so will that be enough time for this to occur? So how would these mutations occur? So the neo-Darwinist position states that we are the result of a series of random point mutations of DNA in the gametes. And so gametes are our mature sex cells, and that unite to form a mutated zygote that grew into a mutated offspring. So what we have here, so we have the sperm and the egg come together to make a zygote. So we need to have mutation in our gametes, in our mature sex cells, and when they're mutated, they come together to create a mutated zygote that's going to grow into a mutated offspring. Then nature is going to select for or against the change expressed by the mutation, depending on if it's beneficial to the environment that the offspring is currently living in. So if selected for like that mutation is a positive thing that's helpful for the offspring in the environment, if selected for, through many generations of interbreeding, the beneficial mutation gradually becomes a dominant trait in the improved herd. So if it's a beneficial mutation, then that offspring will thrive and do really well, and then it will have more offspring, and then that trait will become now a dominant trait. Everybody will kind of have it. So this random plus selective process. So the idea is it's random because it's a random point mutation, but it's selective because nature kind of selects. Is this a positive mutation or a negative mutation? Is how you can think about it based on the environment that the offspring is in. So it's a random plus selective process was repeated a myriad of times for the needed million mutations to occur to reach the present. So that's what has to happen for us to have these mutations kind of occur and be dominant traits. So I want to just be clear about a few assumptions that would be made in the calculations. So first, the mutations at the level of DNA are random. Now this is important because if you know, say you're like, okay, I have this and I want to mutate to get this, then you actually are having very specified mutations because you're trying to get to your end goal. But natural selection says we don't know what the end goal is. Natural selection is just saying it's mutating randomly, 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 and then we get this end goal. And that it just happened to mutate randomly and got hominids. And so we have to kind of do the same. And actually, this is more in favor of evolution because if you have very specified mutations that you're trying to get to a goal, it's actually harder to do that than just random. So it makes the calculations more in favor for evolution. So the mutations at the level of DNA are random, and this also fits natural selection's theory of how it works. And then it's going to be selected for by nature's challenges. The reason that we're saying that is because if it were selected against, then obviously it's going to take even longer to mutate and for you to get the evolution at the end result. Because if there is something that's the mutation is against and it's not very beneficial in nature, then that offspring will die off and it will not become a dominant trait into the herd and it will take even longer. Also, if it's just a neutral mutation, it's not beneficial, then kind of the same thing goes. It kind of slows down the evolutionary process. But if it's selected for, then it's going to go into the herd and be a dominant trait much faster. And so remember, we're going to make all the assumptions to make the evolution kind of as probable and as quick as possible. So we're going to select for by nature's challenges. No mutations are fatal. So this is what I was talking about in the sense of not selected against. So no mutations are fatal, so we don't lose any offspring. Anybody that has a mutation, it's a positive mutation, and then they survive and have offspring and continue. So we don't really waste any time in the evolutionary process. And once a beneficial mutation occurs, it cannot be lost by subsequent detrimental mutations. So what happens is you could have a mutation that's really great, and then you have offspring, but now this offspring has a new mutation that causes it to die. And so because it caused it to die, it loses that beneficial mutation that we needed to kind of go throughout the herd to become a dominant trait. And so what we're saying is we're preventing any of that loss from happening. So, like I said, all these assumptions are actually in favor of the Adam out of ape theory, meaning it's the best possible probability. So, we're looking at the best option for evolution to occur. So, even though all these assumptions are highly unlikely, we have seen this in just nature. Not all mutations are beneficial. Some of them do cause fatality. Sometimes you have a beneficial mutation, but then it gets lost due to environmental change or other mutations. And so it's not very likely, but we're going to go with it for the math. Okay, so let's talk about mutation rates. So in human cells, the maximum mutation rates are 1 per 10 cell divisions. So it's a pretty high mutation rate. But in gametes, the mutation rates are much lower. So 1 per 10,000 to 1 per 100,000 generations. But interestingly, this lower rate is actually not unexpected. So gametes are the source of the next generation. This is where all the data, all the information that you have that's going to determine the offspring is at. So these gametes need to really protect against mutations. So they have a lot of biological machinery that kind of guards against these mutations. And they have a lot of different lines of defense. So if mutation gets through the first line of defense, there is a second one. So that's why the mutation rates are a lot lower than in just regular human cells because we have so many regular human cells versus our sexual mature cells that if we have a mutation, it kind of doesn't impact us as much as if it were in the gametes. But for evolution to occur and to have this mutation into the to be a dominant trait in the herd, then we need it to be in the gametes. So now let's talk about breeding rates. So we not only need the mutation to occur, but then we need it to spread within the herd and we need it to, continue from generation to generation. So approximate breeding population for herds is about 10,000 members. This is a scientifically kind of determined number based on larger mammals. And so that would include hominids and our ape-like creatures. We're going to assume a yearly mating season and that all sexually mature members participate. So even though in real life, maybe not everybody is going to participate and not everyone would then, have offspring every year. But we're going to assume in this case that that's the case. So we're going to assume a gamete mutation rate of every 1 per 10 matings. And so if we kind of go back, it's every 1 per 10,000 generations or 1 per 100,000. And so we're going to go with the better mutation rate. So we're going to assume a gamete mutation rate of every 1 per 10 matings. So if we have 100,000 members in our herd, then that means 5,000 of them, because it's pairs for the herds. 5,000 are going to be in the matings. And so every 1 per 10 matings would give us 500 matings in the herd every year. Mutations, my apologies. So 5,000 members active. And in those 5,000, 1 per every 10 matings gives us a mutation, 500 mutations in the herd every year. Okay, and so remember 3% is occurring in the express region in the genome. So 3% is the active part. So 3% of 500 mutations in the herd would then be the 15 that we need. because it could be that the mutation occurs in the inactive, in the part of the DNA that is not involved with the encoding of the proteins. So we need it to be in that 3%. So 3% of 500 gives us approximately 15 mutations. And there are four possible nucleotide bases. So A, T, G, C. And on average, only one in four actually lead to positive mutations. The other ones will either be neutral or detrimental. So thus, of the five mutations, only four, approximately four mutations will remain. So now let's talk about the time for the mutation to spread and become dominant. So we have the mutation, but we need this mutation to not only just go one generation, but we need it to spread out. So if the mutation is neutral, it's going to take 5,000 generations to be a dominant gene. And this is just common genetics here. So I have a lot of my references at the end of this talk so that you can see where these numbers are coming from. However, if the mutation provides a 1% advantage, which you might think 1% is not very high, but it's actually a very generous percentage that it would be that advantageous. Most of the time a mutation isn't even at 1%. If it's not neutral, it's not that much advantageous. So say if a mutation provides a 1% advantage, then we would only need 500 generations. And so remember we said that we're going to pick the data that is actually the most helpful, that's in favor of evolution. So in this case, we're going to go with the 500 generations because we're going to assume that these mutations are favorable mutations. And so we'll go with that for it to become a dominant trait, we need 500 generations. Okay. And so we need 500 generations for that one mutation to become dominant. Now, we want to assume that the common ancestor becomes sexually mature at around 7 years of age, which is similar to our modern day chimps. So, if we want to think about this, how long would it take? So, 500 generations is what's needed for that one mutation to become a dominant trait. And if we have to wait for them to be about 7 years of age before they can be sexually mature, this is going to take 3,000 years. So you can kind of see in this picture, originally the traits, the mutation, which is kind of that orange color, is not present. But then over time, as it kind of goes throughout the herd, it's going to kind of spread and become more dominant over that 3,000 year time period. So one mutation takes a little over 3,000 years. So then the question is it possible for the 1 million mutations, because remember we need 1 million, to occur in the 7 million year time frame? So one mutation takes 3,000 years and we need 1 million in 7 million. So if we assume that the mutations could occur in any order, so this is also another assumption that is helping it, because you might say, well, we need the hip bones to evolve before the leg bones can evolve or something of that nature. And so there would be then more specificity required in those mutations, which actually makes it more complicated and harder to do. So instead, we're just saying we need 1 million of these, and it doesn't matter in what order they occur. So it makes it more favorable for evolution. Okay. However, only 15 traits can concurrently be altered. This is another kind of just one of those facts from genetics, that the max amount of mutations that you can kind of do all at the same time is 15. So only 15 traits in our genetic code can be altered at the same time concurrently. So we have 90 million active bases where mutations can occur. So remember, 90 million places where these mutations can occur. And there is a 17% chance a mutation will occur in a beneficial site. Okay, so remember that we only have 3% of that of the DNA is active for protein coding. The other 97% is not. and only 1 in 4 chance that the mutation will be in a beneficial base for that site. So remember, in each site, it could be an A, T, G, or C at that site, and only one of those 4 actually lead to a beneficial mutation. So 17% chance. So if we want to look at the probability of success, so 17%, 0.17. And then our 1 in 4 chance that it's going to be at a specific base site is 0.25. So we do the 0.17 times the 0.25 and we get 0.0425 is our probability of success that that mutation would occur in the right location. Okay. And then probability of failure is 1 minus the probability of success, 1 minus p. So 1 minus 0.0425 is 0.9575. So right now my apologies. Right now the probability of success is actually very, very small in comparison to the probability of failure. So let's just continue to look at the statistics. So the top part here is just for the first generation. So what is the probability of success of that mutation of it happening in the right part of the DNA, in the right base, and that it is actually in a favorable consideration? It's 0.0425, and that's just for the first generation. Now we need 6 subsequent generations. So the probability of success in subsequent generations is determined by this equation. So the probability of success is 1 minus the probability of failure raised to r. And r is the number of generations needed. So it will take 40 generations to occur before there is an 82% probability that any one of the potentially useful mutations have occurred. Okay. And that's kind of like when we get to 82%, it kind of starts to just plateau out. And so that is kind of like our goal point is this 82%. So it's going to take 40 generations before we get to that percentage that any useful mutation has occurred. So what does this mean? It means that we need 40 generations before one mutation becomes dominant. So let's just go back and look at that. So 40 generations before we hit that 82% probability. And that's just for one mutation. Okay. So 40 generations for one mutation. But remember we said we had a million mutations that need to become dominant. So for a million mutations, then we would actually need 40 million generations. So it's going to take 40 million generations for 1 million mutations to become dominant. Now, remember the other thing that I actually didn't even include in this math, because it even makes it even longer, is how long it's going to take for it to spread throughout the herd. So we just talked about how much it takes to be dominant in kind of going through, but not throughout the herd. So remember we said it takes between 50 to 5,000 generations for each mutation to spread throughout the herd versus just kind of one generation, the offspring, etc. So the fossil record, remember I said only gives us 7 million years. And so based on this, we have 7 million years, but based on the math for genetic evolution to occur, it would take 40 million years to get that 1 million mutation. So what we're looking at here the question is there a common ancestor between chimpanzees and hominids? Because this is the big kind of question, and this is the big thing that Darwinian evolution says. Darwinian evolution says that you kind of have the ape-like creatures that then become hominids and then to modern man. And so our question is, if all of that evolution could occur, is there enough time for all of that to happen? And the question here that we looked at is there's not. There's not enough time in the fossil record because we only have 7 million years and we actually really need 40 million years for this to occur. So thank you so much for attending my talk. Here are just a few of the references related to where some of the data points come from and Um, Something that you can use to kind of just dive in deeper if you would like. And so I hope that you join me for my next talk. We'll now start to actually look at things that are shared in the Bible that have a scientific perspective. So far in my Science and Religion series, we've looked at things in the scientific field and how does that relate to what the Bible says. Now we're going to look at what does the Bible say that has some scientific implications. And so I hope you join me for my next talk as we kind of switch gears and look at that.